Hello, my dear students of class 11 science. Welcome to biology class. So we'll continue with the chapter anatomy of flowering plants. In the last class we have learned about the anatomy of dicot and monocot stems. So in today's class we'll learn about the anatomy of leaves. Okay, it's in the page number 6.31. So today we are going to learn about the internal structure of dicotyledonous and monocotyledonous leaf. So first of all, we'll begin with the internal structure of a dorsiventral or dicotyledonous leaf. Now, what does dorsiventral leaf means? Okay, so mainly the leaf of the dicotyledonous plants are mainly dorsiventral. That means only the one side of the leaf will get higher amount of sunlight, or it will receive higher amount of sunlight compared to the other sides. Clear? So therefore the leaves of the dicotyledonous plants are arranged in a horizontal position, that is at right angle to the rays of the sun, so that the upper surface of the leaf will receive more amount of sunlight than the lower surface. Since in such type of leaf only the one side of the leaf is getting a higher amount of sunlight compared to the other side, it is known as dorsiventral leaf. Clear? Okay, so when we go through the thin section of this dorsiventral leaf or the dicotyledonous leaf, while passing through the midrib, we can see the following structures. Okay, we can see the presence of upper epidermis. Okay, we can see the presence of lower epidermis. Now the upper dermis is covered by a thin layer of cuticle. Clear? And the lower epidermis, it may contain cuticle or cuticle may be absent. Clear? Then we will see the presence of mesophyll cells. So between the upper epidermis and the lower epidermis, we will see the presence of mesophyll cells. Clear? And mesophyll cells are divided into two types. First one is palisade mesophyll cell and second one is spongy mesophyll cell. Alright, then we will find the presence of vascular bundle, that is xylem and phloem. Okay, so you can see the presence of xylem, you can see the presence of phloem, and these vascular bundles are surrounded by the bundle sheath. Clear? So let's see the structure of the dicotyledonous leaf or the dorsiventral leaf. Okay, so first one, upper epidermis. So that means we are going to learn about this structure. Okay, this cell. So we are going to learn about this particular structure, which is called the upper epidermis. So this upper epidermis, it consists of a single layer of parenchymatous cell, that is parenchyma cells. Right? Now the outer wall of these epidermal cells, they are mainly covered by the cuticle. And if you remember, we have learned in class 10 during photosynthesis and also during transpiration the function of cuticle. Okay, the cuticle is to be thick in order to prevent the loss of water through the process of transpiration. If you remember the thick cuticle over there, again transpirations. So the outer wall layer of the epidermal cells, they are covered by the cuticle. Okay, and epidermal cells, they do not contain chloroplast and they do not have stomata. So here you can see in the upper epidermis, we don't see the presence of the minute pore or the openings, which is called the stomata. So stomata and the chloroplast are absent in the upper epidermis. Now this next part, which is called the lower epidermis over here. So now we are going to learn about this structure, okay, which is called the lower epidermis. Clear? So it's also a single layer of cells which is present on the lower surface of the leaf. Now in the lower epidermis, I've told you that the cuticle may be present or cuticle may be absent, and if the cuticle is present, it will be very thin. Okay, and it contains numerous stomata. So lower epidermis contains numerous stomata. So you can see in the figure also that this structure, the small opening, is called the stomata. You can see the word stoma. So the single opening is called the stomata. Clear? And each stoma, it consists of a minute opening that leads into a larger air cavity. Now what is the function of stomata? Okay, so stomata will help in the gaseous exchange between the atmosphere and the leaf tissues, and also help in the evaporation of excess of water from the leaf, because too much of water is also not so good for the plant. So those excess water will be lost in the form of vapor by the process of transpiration through the help of the stomata from the surface of the leaf. Okay, so we have just finished about the upper epidermis and the lower epidermis. Now let's move to the next part of the dicot leaf, that is mesophyll. Okay, so this particular mesophyll tissue is present in between the upper and the lower epidermis. Now which part is upper epidermis? So this part is the upper epidermis and this part is the lower epidermis. Okay, so upper epidermis contains the cuticle, whereas in the lower epidermis the cuticle may be absent, or if it is present it may be like it is very thinner. Okay, so between the upper and the lower epidermis we will find the mesophyll tissue. I have told you that in between these two structures, the upper and the lower epidermis, we will find the mesophyll tissues. Okay, so these mesophyll tissues, it consists of chloroplast containing the photosynthetic tissue. That means it will help in photosynthesis. So the mesophyll forms a compact mass of cells around the veins or the bundles. So these mesophyll tissues or the mesophyll cells is divided into two types: palisade mesophyll cells and the spongy mesophyll cells. So let's see the palisade and the spongy cells. Okay, so first one is palisade parenchyma. So this palisade parenchyma or the palisade mesophyll is close to the upper epidermis. You can see over here the palisade mesophyll, and this is the upper epidermis. So this palisade mesophyll is close to the upper epidermis. Okay, and cells of this palisade like palisade mesophyll cells are generally elongated cells. Clear? And here we can find few or no intercellular spaces. That means the space between the two cells. So here the intercellular spaces may be few in number or it may be absent, but it contains a large number of chloroplasts, and since it contains the large number of chloroplasts, it will help in the process of photosynthesis. And all of you know what is photosynthesis, right? Okay, now let's go to the spongy parenchyma. So spongy parenchyma is close to the lower epidermis. So here you can see the lower epidermis, and close to the lower epidermis you can see the presence of spongy mesophyll cells. So it lies towards the lower epidermis. So palisade mesophyll lies to the upper epidermis. The cells are elongated with few or less intercellular spaces, but they contain chloroplast and will help in photosynthesis. Whereas the spongy mesophyll cells, they are close to the lower epidermis, and the cells may be round, may be oval or irregular and thin-walled parenchymatous cells. So here you can see the structure. So this one, from here to here you can see the spongy mesophyll cells. So the cells may be round, it may be oval or it may be irregular, and they are thin-walled parenchymatous cells. And here you can see there are presence of large intercellular spaces. These are the intercellular spaces. Okay, so cells are loosely packed, and we can see the presence of large intercellular spaces, and we can also see the presence of air cavity. Okay, and mainly in the lower epidermis we can see the presence of stomata. Also we have learned that thing. Okay, so what we can see over here is that this stomata will help in the exchange of gases. So since it is present in the lower epidermis, here also we can say that the spongy parenchyma will help in the exchange of gases and water vapor through the intercellular spaces and the air cavity. That means from here through the spaces and through the air cavities there will be exchange of gases and water vapor by the help of spongy mesophyll cells. Okay, and this spongy parenchyma will also help in the manufacture of food. So don't get confused. That is palisade and mesophyll cells are divided into two types: palisade mesophyll and the spongy mesophyll. Okay, now next one, vascular bundles. So vascular bundles is mainly composed of xylem and phloem. So you can see the presence of xylem and you can see the presence of phloem. So xylem and phloem, they will constitute the vascular bundle. So xylem lies towards the upper epidermis, let's say, and it mainly consists of vessels, tracheids, xylem parenchyma and the wood fiber. Whereas phloem, it lies toward the lower epidermis. It is composed of sieve tubes, companion cells and phloem parenchyma. Okay, so xylem lies toward the upper epidermis and the phloem lies toward the lower epidermis. And xylem is mainly composed of xylem vessels, tracheid, xylem parenchyma and the wood fiber, whereas phloem, it lies toward the lower epidermis and is composed of sieve tubes, companion cells and the phloem parenchyma. Okay, now these vascular bundles are surrounded partially or completely by a thick layer of screening chimerous cells, which is called the bundle sheath. So here we can see structure. So this one is our vascular bundle. Okay, this is the vascular bundle, and inside you can see the presence of xylem, you can see the phloem. Now this vascular bundle is surrounded by a sheath which is called the bundle sheath. Clear? So these structures are present mainly in the mesophil part of the leaf. Okay, so in mid-rib the vascular bundles are conjoined and collateral. Phloem lies toward the lower side and xylem lies toward the upper side. So here we have finished the internal structure of the dorsiventral leaf or the dicotyledonous leaf. Remember, we have got upper epidermis, we have got lower epidermis, we have got mesophil tissues or mesophil cells. We divided into palisade and spongy mesophil cells. Then we have got the vascular bundle. Now we will look into the structure of the monocot leaf or the isobilateral leaf. Remember, dorsiventral leaf will only receive the sunlight on one side compared to the other sides. Clear? And mainly in such leaf, stomata was present only in the lower epidermis. Upper epidermis was devoid of chloroplast and is devoid of stomata. Now we will move into the monocot leaf or the isobilateral leaf. So isobilateral leaf used to receive the sunlight on both the sides. So both the sides are equally exposed to the sunlight, or they will receive the light equally in case of isobilateral leaf or the monocot leaf. Okay, now when you go to the transverse section of the monocot leaf or the isobilateral leaf, we can see the presence of epidermis. We have got the upper epidermis. Okay, we have got the lower epidermis. We will see the presence of mesophil tissue, and we will see the vascular bundle, that is xylem and phloem. So let's go in detail now. Okay, so epidermis. So when we look into the structure of the monocot leaf, there is no such difference in the upper epidermis, and there is no such difference between the lower epidermis also. So we don't find any differences between the structure of the upper epidermis and the lower epidermis in case of monocot leaf or the isobilateral leaf. Okay, because stomata used to occur on both the surfaces. So here you can see the presence of stomata. Okay, so this is the stomata which are present on both the epidermis, on the lower epidermis as well as on the upper epidermis, and these stomata are mainly surrounded or protected by the guard cells. Clear? So leaves with stomata on both the surfaces. Since the upper epidermis and the lower epidermis contains stomata, so such types of leaves are called epistomatic leaves, because both the surfaces, that is upper epidermis and the lower epidermis, contains the stomata. So such types of leaves are called epistomatic leaf. And the epidermis is made of compact cells which lack the chloroplast. Now in grasses, look very carefully, in grasses, certain cells of the upper epidermis, okay, along the veins, they are modified into large empty colorless cells, and such cells are called bulliform cells, bulliform, bulliform cells. So when these cells will absorb the water, okay, when these bulliform cells will absorb the water, they become turgid and they usually open up, they become exposed. But when they become flaccid, when they lose the water, when they become flaccid, okay, the leaf will curls inward to minimize the water loss, because you have learned in class 10 about the turgidity and the flaccidity of the cells. Okay, so here when this bulliform cells will absorb the water, it becomes turgid and the cell becomes exposed. But when it becomes flaccid, it will shrink, or it will curl up and it will close. Okay, next one is the mesophil cells. So in monocot leaves, the mesophil tissues is not differentiated into the spongy and the palisade parenchyma cells. Clear? Like in dicot, we are like the mesophil cells were divided into the palisade and the spongy parenchyma cells. But here in monocot leaf or in isobilateral leaf, the mesophil tissue is not differentiated into the spongy and the palisade parenchyma cells. And these cells usually contain the chloroplast that will help in photosynthesis. Okay, so we don't see the differentiation of mesophil tissue into spongy and palisade parenchyma in the monocot leaf. Next one is vascular bundles. So in monocot leaf, a large number of closely placed small and a few vascular bundles occur. So you can see over here, you can see a closely packed small and a few vascular bundles. So here you can see the presence of vascular bundle, This one is vascular bundle. Even here we can see the presence of vascular bundle. Okay, so these are conjoined and collateral, the meaning of which I have learned already in the previous classes, so try to recall it back. Okay, and this vascular bundle is mainly composed of xylem towards the upper surface and the phloem towards the lower surface, and due to the parallel venation in monocot leaf, it reflected in the similar size of the vascular bundles. So here we have finished about the structure of the dicot leaf and the monocot leaf. Now from your textbook, kindly go through the page number 6.33, table number 6.23, page number 6.33, table number 6.23. You will find the table where you will see the differences between the dorsiventral leaf and the isobilateral leaf. Then we have got one particular term called the Cranch anatomy. Okay, that means in certain cells, okay, certain leaf, the upper epidermis becomes bulliform. Okay, and here the undifferentiated mesophyll cells, that is the cells which are not differentiated into palisade and spongy cells. So those undifferentiated mesophyll cells, they form a ring outside the bundle sheath. Okay, listen very carefully. The undifferentiated mesophyll cells form a ring outside the bundle sheath, and this arrangement of the mesophyll cells is called the Cranch anatomy. So kindly go to the book, you will find this term. Okay, so here we have finished the anatomy of the leaf also. So this much for today. In the next class, we will begin with the new topic. We will learn about the secondary growth in the stem. So this much for today. See you in next class. Thank you.